Hi, I'm Meredith Blackwell, and this is Don Pfister, and we're part of the Oral History for Mycology project. We're at the University of Florida on their campus and in the campus hotel, which is a Hilton. And it's the 11th of July, 2022. And we're here to interview brothers studying mycology. And so Andrew Mann and Michael Mann are students, they're PhD students, and they're in different universities. So we'll let them introduce themselves and tell us where they are and what they're doing.
So I'm Andrew Mann. I'm a PhD student at the University of Minnesota, and I'm advised by Dr. Blanchett, and I study beetle-associated fungi.
I'm Michael Mann. I'm a PhD student at University of New Mexico. I'm co-advised with Lee Taylor and Jennifer Rogers. I study plant-associated fungi and developing molecular methods.
So Bob Blanchett had suggested it might be nice to interview the brothers together. And Don and I have been trying to think of other sibling pairs that have been mycologists. And Don Wicklow, who was at Peoria at the USDA facility, and his sister, who was at Boise State for her career, Martha Wicklow Howard. And then the other pair that we think of are the Raper brothers. John Raper was at Harvard. He studied fungal genetics. And his brother, Kenneth Raper, was first at the USDA in Peoria, and then later went to the University of Wisconsin. And they both have published very, very much, and they're both distinguished mycologists of the Mycological
They both
Society.
had the same advisor.
That's right.
Yeah, their advisor was William H. Weston, who was the teaching award for the societies named after him. Cap Weston was his nickname, and but they were both with him at Harvard as a PhD.
So you're different because you chose different
You're
places.
at different places.
Yeah.
Do you know any other siblings in mycology?
That I can
Never
think
thought of it.
Yeah.
Not in the same field. I feel like there's a lot of people, siblings, but then one's in like statistics or another STEM field, but not in the same discipline. Yeah.
So one of you or both of you chime in about where you were born, and you might have been born in different places since you're not twins, and your early family life, and first time you saw fungus and remember about it, and all that sort of stuff, if it happened.
Yeah.
I never saw a fungus till grad school, or noticed one.
Yeah, so I was born in Virginia, and Michael was actually born in Florida.
Clearwater, yeah.
And we moved around some in our early childhood, but I consider myself to be from Austin, Texas. That's where we spent a lot of our early years, and that's where I went to high school. That's where we both went to high school.
Yeah.
That's where I went to grad school.
Okay.
Who's the oldest?
I'm older.
Okay. I thought maybe because you were talking, you were the oldest.
Oh.
That there's a pattern here.
Yeah. My story starts earlier. But yeah, I mean, early growing up, we spent a lot of time outdoors, hiking and trails. Our mom studied biology for her undergrad and was very interested in biology and ecology. And she was a high school teacher while we were growing up. And so she's also very interested in education, and that's really important to her. So we spent a lot of time in the trails, there were some canyons near our house. And I just really enjoyed nature. I don't know how you felt about all of that early childhood.
Yeah, I don't think we had much exposure to fungi at all. I mean, we'd go to West Virginia, but I don't think we were there during the peak
I remember
seasons. I don't remember much as a kid.
Yeah, I would say, so our grandparents are from West Virginia, and they own some tree farms, and that's actually, so I studied forestry for my undergrad, and that's how I got exposed to forestry and interested in the potential of becoming a forester. But I remember one time, probably 10 or so, we were in West Virginia, probably around this time of the year, and we were with Walt Shoup, and so he was the park superintendent of one of the state parks in West Virginia, and very good friends of our grandparents. And we were driving, and we found some chicken of the woods on a tree, and ate it that night. And I thought that was super cool. That might have been my first experience with mushrooms and fungi. But yeah, I mean, growing up in Austin, I never thought that I would become a mycologist.
And did you have anything to do with university? Where high school trips did you go over to the university and meet any of the biologists there?
We did. Our mom took us to this. It was like Friday night science talks at the university, which I really enjoyed. I didn't understand a lot of. It was more for the public, but I still didn't understand a lot of what was happening. But I liked being there. I think you had.
I had much, yeah. So we did a lot of science fair projects.
Yeah, that's what I was wondering. Yeah.
And through that, I mean, we did it. I don't know if you did every year, but through middle school through high school, we dabbled in a bunch of different projects. So like I did an early one culturing probably bacteria, fungi from like cockroaches for fun, and just like putting them on a petri dish and letting them run around and see what grew. And then I got more interested in doing plants work, and I really enjoyed that. And I, from doing all the science fairs, we saw that like a lot of other students were working with like universities. So I started just emailing, and I just happened to email Christine Hawks. And I knew probably nothing about fungi at that time, but I was interested in like plant microbe interactions from doing the science fair projects, and just stumbled in that lab. And thankfully, Ben Sykes was there to help and guide me a lot through it, and really sparked my interest in mycorrhizae, endophytes. And so, but it's funny going back, I don't think in high school there was any education on fungi. I found some of my notes, and it was like my AP bio class, there was maybe 3 sentences I wrote saying one characteristic of Ascomycota was like the extent of what we covered. And I think, when I applied, I think Ben asked me if I knew anything about mycorrhizae, and I think, thinking back, I think I confused it with rhizobia. And I was like, oh, I think we learned about in school. And then later I was like, wait a minute.
At least you didn't have to tell him that you'd made a mistake.
But now it's on video.
Oh.
Now it's in the record,
Yeah.
yeah.
But
Yeah, it's funny. I never noticed a fungus growing up, never. And I was in the woods all the time. And when I was quite young, I just don't remember seeing one. Ever.
I do.
Yeah.
Yeah, I have memories of seeing fungi in the yard and on the farm and so forth.
So we kind of have that in common, except for the chicken of the woods.
Yeah, that was kind of my only early memory.
So it's so curious with the background of very little fungus activity and so forth that the two of you end up doing mycology. Did you talk about this?
No, it's a coincidence, really.
So where were you undergrads?
I went to Northern Arizona University in Flagstaff.
Yep.
I went to University of Colorado, Boulder. Yeah.
And so when I was in Flagstaff, I became really interested in forest pathology as well as forest entomology. And I worked some with Rich Hofstetter, who's the forest entomologist, and really interested in bark beetles and some other insects that cause a lot of damage to the trees. And I was really interested in like the symbioses that were happening with not only with fungi, but bacteria as well as mites are carried on these beetles. And there's even, I studied a moth that is associated with this virus. So I think that's kind of how I got into mycology, is from entomology, and these beetles and other forest insects have such important relationships with fungi, and fungi are really helping the beetles out probably and helping them attack the trees.
So what sort of fungi are you isolating?
Or are
with
you
my
isolating?
PhD, yeah, I'm isolating, culturing a lot of fungi. Really a lot of things. So the ophiostomatoids are what we think of and as being obvious symbiotic fungi with the bark beetles.
So are they opiostoma and ceratocystis lineages, or you said opistomatoid. So which ones? in
Most of
particular
the genera I get mostly are Ophiostoma, Leptographium, Grossmania, mainly those types of. And it's tricky because they're always changing.
Yeah, yeah. I was at a meeting one time, and I was told that, well, I saw a poster. There was a dog that was trained to find some of these leptographiums, and it could identify 4 species. And the 5th behavior it did was if there was a combination. So I thought it was pretty neat. The mycologist, redundant? No. But I think there are many other fungi the dog probably couldn't identify. But it was interesting.
That's interesting.
So they must put out an odor that probably attracts the beetles.
I think so, yeah. Mm-hmm.
Yeah. So do you
Michael,
isolate?
yeah, you were at Colorado, so how did that play out here?
So I mean,
That you
from
found?
working in Christine's lab, it was like I came in being like, okay, I want to study plant microbe interactions. I really enjoy. I think it's a fascinating world. And from there, I felt like from my background, I wanted more plant background, so I worked with some more kind of plant focused labs. And it was my degree was in ecology evolution, which was 80% ecology. And so I worked a lot with Tim Seastead. We were going up to NIWAT, up to the LTER. And then also later on, I worked a small amount in Noah Fier's lab. And so it exposed me to a lot more of the next generation sequencing. And
Yeah, I ran into his work because of work he was doing on indoor molds. And did you work on that project?
no, I was working with one of his grad students. I was really curious about endophytes, and so I was, just because I always felt like there wasn't as much attention on them as opposed to other ectomycorrhizae, which now you look at the MSA book and I'm like, well, there's there's a lot of people working on endophytes. But when in college, then I was like, oh, I would be really curious. So I spent time like pulling out some of the common dark septate endophytes and trying to run them through, because Noah had like a server of all of his sequences. So I would just blast them and just see, and it would blow my mind. You go at the Kansa Prairie and half the sequences come back. Maybe some of these DSE species, and just God, they're all over the place, but we know so little.
It was funny. I didn't know about endophytes until one of the guys I was in grad school with went off to Oregon, George Carroll, and started finding endophytes. But at that time, and that was in the mid '60s. Yeah, I had not heard of them.
Well, I don't know that they were commonly thought of even,
No.
and even further on, later on in the 70s and so forth, there was always the debate. Is this real somehow? And aren't they contaminants? And what are they doing? Can we believe this?
And then the grass endophytes, it was even later when they figured out that they were killing or poisoning cattle.
Yeah.
Yeah, and that was pretty late. I don't remember the exact date, but it was bacon and some of them.
But it's a real evolution of the kind of concept of endophytes. When you hear Betsy Arnold talking about this, this is really sophisticated now in what
and
we
it's
know.
happened very fast since that time.
Yeah.
It's molecular techniques instrumental in that group, or is it?
That's the
every part of mycology.
Yeah.
Mycorrhizae for sure.
Well,
and the things you're getting, they're hard to identify if you don't do DNA.
Right. Yeah.
and you miss things. Sorry.
But
Our
even,
morphological characters.
And even if you can culture, you get something, and with these endophytes, you've got, hyphae often. You don't have sporulation. And what would you do in the old days with a fungus like that?
Mycelia sterilia.
Yeah, you couldn't do anything much with it. You could maybe do some physiological tests or try to look at a little bit of function maybe of what they were doing, but it's means now that you can do this. And one of the papers that Betsy mentioned is the Pfizer, which is the group that I've worked on all my career. But because of the sequences, we know now that they're pretty common. There are certain families of the Pfizer are very common as endophytes or endolycanic. Yeah. And there's no way you would have known that. Right. So you're at a great point here to be undertaking your studies.
Mm-hmm. I love when she mentioned it, because I was thinking back. So a lot of my work is done at the Rocky Mountain Biological Lab. So we've sequenced a lot of the grasses and the roots and leaves. And I think like our second most common OTU is Morchella. And I always wondered about that, because like,
oh, I know.
it's just.
So have you run into Carol Boggs?
No.
Okay.
Oh, yes, I know of
The
her, but
butterfly,
yeah.
Well, actually, it's amazing because I work out of her lab now because I've retired and I'm in Columbia, South Carolina. So they let me put my microscope in the lab. And so we've done actually a few projects with students, but not a whole lot. But we have a paper about ready now that we've got a bacterium identified. It took us months. But I used to work a little bit because of a student called Stephen Kassar with some of the bark beetles, and it was really interesting because with the DNA sequencing we could identify them quite easily before waiting to try to do the morphology, which is really difficult with these. Mm-hmm. But I like the way you said opiostomatoid fungi, because one of the things that Stephen showed was that the anamorph kind of followed the tendency of the teleomorphs to mimic each other. And so it was really kind of interesting to find out some of the Ambrosiellas, I guess is a genus, that some of them were opiostoma types and others were Ceratocystis types. And pretty neat.
So how are you projecting your careers now? You're in graduate school. How many years? What year and how long yet for your PhD?
I'm further along. I just finished my 5th year. So
5th year.
You
So
start
you're
wrapping up.
Closing in on it. Closing
It's
in.
time to time to do work.
Yeah.
Have you got your thesis started?
yes, I'm working on it.
And I'm in my 2nd year of my PhD. So I took a little bit of a
one
different path.
weird-
I'm older,
But you did a master's prior, so.
I did a master's, and I spent some time doing things like working for the Forest Service and moving around the country, doing seasonal jobs, sort of thing. But for where I see my career going, I love research, and so any way that I can be doing a research as a professor, or I think there's a lot of opportunities with the Forest Service. I think my interests are much more applied than Michael's are. And I don't know, I like to work with pests or things that are causing damage. And
Well, I've been saying the last couple of months, I have never seen more job ads from the Forest Service than ever in my life.
I think there's something like 5 or 6 forest pathologists right now, which is
one in
unheard
West
of.
Virginia, I think.
Okay.
But anyway, I used to be part of because of the bark beetles, they've still got me on their list, so I got all these ads. But when I was coming up, I would love to have had an all-research job with the government. But they used to hide their announcements and didn't advertise them widely. And then you'd find out that you could only telephone in like for 5 minutes late on Friday afternoon. That's when it was open. Otherwise, the whatever they call it, their bulletin board was closed. So it's a better time now if you want that kind of job.
Yeah.
Which is good.
Yeah.
And you want?
Yeah, and I'm open. I've been having I've really enjoyed like my work on kind of trying to place. I'm doing a lot of work just sequencing longer reads and trying to place them onto phylogenies and because a lot of the plant-associated fungi just don't seem to be well represented in any of the databases, or just there's so much. So it's like you do all these like huge Illumina sequencing next generation kind of runs, and you can't identify much to beyond phylum. And it's very disappointing. You get there and you're like, we just know we have a lot of fungi in our plants or soil, but you feel you
Do
hit.
you work much in the desert? Do you get in desert house?
Yeah, I work somewhat. My other advisor, Jen Rogers, runs the Sevietta, so we do a lot of work on the different grasses down there. And
I used to go down and work with Bob Gilbertson out of Tucson, and we were studying slime molds mostly. But
Oh, cool.
you know, he did wood decay fungi, so we did those too. It was really it was an eye opener what's out there.
Yeah.
20 degrees cooler under a saguaro than out in the sun.
Yeah.
I wish I could have gone to see some of the Neurospora after some of the New Mexico fires. Don was telling us stories about how just blankets the trees, and it's remarkable.
Supposedly you can see some of that in Louisiana after fires. They burn the cane, or they used to, but I think some people probably still do it. The cane stubble after the harvest and sugar cane. Anyway, there's supposed to be a lot of it there. And I think a whole lot of the things in the Rossford collection have come from Louisiana because
Yeah,
of
well,
that.
they're looking at wild Neurospora much
Yeah.
more now than used to be, but yeah, I've seen it in the tropics on burns.
So you want a teaching job?
Yeah, I want to continue with research in some capacity. Yeah. I don't know exactly which path to go on, but I really enjoy especially the bioinformatics and that side, and I like kind of the working with bioinformatics, ecology, mycology, and how they all intersect.
Yeah, and you're in a good place for that with the work that that I know that Lee's been doing. I'm more familiar with his earlier work in the Arctic.
But yeah, I think I'm going to probably, that's one of my chapters I'm hoping to work on, is looking at the phylogeographic patterns of some of the samples he's collected up in Alaska, because he's a really cool transect up through Alaska into the tundra. Also this other one going up through Russia. And so I've been trying to look at those samples and...
Don't go to Russia.
Yeah. Well, we already got the sample, so we're...
Oh, okay. Shouldn't say that, but eventually you can go see where they were collected.
Yeah.
One of Don's students, one time, he helped me reenact something that was in one of Lee's papers, and we took, 200 grams of soil and held it out in his, so we could tell how many fungi would be in that. Do you remember when we did that?
Yeah.
You might not have been around.
But yeah, Lee's project is so fascinating because one of the things I've enjoyed most is say I work in New Mexico and I try to look up other sequences of common things, and it's and thinking back of Betsy's talk about how a lot of these fungi seem to be somewhat localized. Most of the top sequences I hit back to work from Don Matvig's student, Andrea Porras Alfaro. It's I just hit the exact things she was finding 10 years before. Or I look at a lot of Lee's, he has a soil data set, it's hitting his other soil sequences.
I've got to talk to Betsy about one near where she works in one of the riverine habitats. Bob Gilbertson and I were collecting slime molds, and we saw this thing on leaves, and it covered an area probably half the size of this room, these fruiting bodies on leaves. And at first we thought it was a slime mold, and we looked at it more closely. No, yes, no. We couldn't really decide. So we took it in and then made them out and saw mycelial structure. So it wasn't a slime mold. But then we couldn't figure out what it was. And so I had left a slide over the weekend in lactophenol cotton blue. And I came back in the Monday and said, "What's on here?" And it was a penicillium. And I thought, what? And then I realized we had a synemedal, I guess it's a Tauromyces now, but it was growing all over the place, and we call it myxomimic. And so Betsy has it in the portal, and so she actually had it as a specimen at the University of Arizona because Gil had deposited it. And lo and behold, she has it as an endophyte on some of the oak trees that had dropped the leaves that
Yeah.
we found.
Yeah.
Wow.
So it was really kind of neat to find that thing.
Yeah. No, it's
And I have to talk to her about it. I haven't ever done it. But it's already been described. Keith Seifert and some Spaniards found it. So they've got a really good description. But it's really funny. So back to the beetles then. For a long time, they were really close to my heart. And then I've gone to other beetles and looked at their gut contents.
Mm-hmm.
And that's mostly Ascomyces yeast, but a few Sphingomyces. But we're not finding a whole lot of filamentous fungi. It's not in the ones we've been looking at, which eat mushrooms. And I think Noah had found some in fly guts, some of the same ones. So it was kind of interesting. So there's these yeast are specific to the mushroom feeders. It looks like different insects that feed on mushrooms. But we've been looking at pasalids now.
Okay.
You probably, do you have them? You'd have them in West Virginia. But
Yeah, I don't know if they live that far north in Minnesota. I think.
I think in eastern Canada, they may go into Canada
Oh,
further
okay.
east where you know,
Yeah.
coastal.
Actually, I did a mini project in my undergrad on them.
On pasalids?
Yeah, and the mites that are
Yeah.
found on them, because they were pretty easy to find. Just turn over a bunch of wood samples and huge beetles.
Yeah.
And
Yeah.
Were they all adults that you were finding?
Mhm.
Yeah. And the interesting thing about them is the Lobulbiniales. They have a group of lobules that are on these pasalids.
Okay.
And so on the mites as well,
Okay.
but
yeah.
they're hard to see.
yeah, I haven't thought about them for probably 10 years, but yeah. And also
You could
before I
go-
knew much about fungi,
You could go back and have several more projects on them.
kept the samples.
They're really interesting. I like insects and beetles especially.
Mhm.
Any more?
I don't think so.
Oh, it's great to have the two of you.
Yeah, yeah. Do you ever think about doing something together?
We,
I don't-
you help me
I
out
help, yeah,
sometimes with
I
my
did them.
analysis, and I think that we could maybe
Yeah,
down
probably
the
do
line.
some. Yeah.
Yeah.
Yeah. I know 3 brothers who are mycologists, and they published a paper together on occasion.
Well, you should do at least one.
Yeah.
Yeah.
Establish yourselves together.
And I
So-
don't know that the Raper brothers ever did that.
I don't think they ever published
yeah,
together.
they were very different personalities.
Well, they
and,
studied very different things.
yeah, both.
Yeah.
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 So,
Yeah.
I think that could
Have
be
to
fun.
come up with something.
Yeah.
Yeah.
Great.
yeah,
Well-
I think you should aim for it.
Thank you for taking the time. For
Yeah.
Thank you. This is
doing
great.
this. It's fun.